Imagine spending over $100 on a custom qPCR assay, waiting almost a week for your primers to arrive, carefully extracting RNA, running your qPCR experiment, getting beautiful amplification curves, only to realize that your primers amplified the wrong transcript. Unfortunately, this happens far more often than most researchers think. Not because qPCR is unreliable, not because the software made a mistake, but because the assay was designed incorrectly before anyone entered the laboratory. The good news: almost every one of these mistakes is preventable. In this tutorial, I'll show you exactly how I design qPCR primers from scratch using completely free online tools. We'll start with nothing more than a gene name and end with a fully validated primer pair ready for ordering. By the end of this video, you'll know how to choose the correct transcript, download the right sequence, design high-quality primers, evaluate primer quality, avoid genomic DNA amplification, and verify that your primers amplify only your target gene. I'm a bioinformatician and the director of bioinformatics. On this channel, I share practical tutorials on molecular biology and bioinformatics that you can immediately apply to your own research. Welcome to bioinformatics. Let's get started. Before we design anything, let's spend 2 minutes understanding what these primers are actually used for. Many beginners hear the terms PCR and qPCR and assume they're basically the same experiment. They're not. Conventional PCR simply answers one question: is this DNA sequence present? After amplification, we load the products onto an agarose gel. If we see a band at the expected size, the answer is yes. If we don't, the answer is no. That's endpoint detection. qPCR works differently. Instead of waiting until the reaction is finished, the instrument measures fluorescence during every amplification cycle. As the amount of DNA increases, fluorescence also increases. The instrument continuously records that signal and generates an amplification curve. From that curve, it calculates a value called the CT, or cycle threshold. The lower the CT value, the more starting template was present in your sample. This is why qPCR is the gold standard for measuring gene expression. In a typical gene expression experiment, we first isolate RNA from cells. That RNA is converted into complementary DNA, or cDNA. Then qPCR quantifies how much of a specific transcript is present. And here's the important part: if your primers amplify the wrong target, your CT values become meaningless. So primer design isn't just another step in the workflow. It determines whether your biological conclusions are correct. Now that we know what we're trying to measure, let's choose the correct transcript. Open your browser and go to the NCBI Gene database. Today we'll use one of the most studied human genes, TP53. Search for TP53 and open the gene record. Don't scroll too quickly. One of the biggest mistakes beginners make happens right here. If you scroll down to the RefSeq transcript section, you'll notice something interesting. There isn't just one transcript. There are several. Why? Because most human genes undergo alternative splicing. That means one gene can produce multiple mRNA transcripts. Different transcripts may contain different exons. Sometimes they even produce different protein isoforms. If you accidentally design primers against the wrong transcript, your qPCR experiment may work perfectly while measuring the wrong biological molecule. So before designing any primers, answer one simple question: which transcript do I actually want to quantify? For most gene expression experiments, the safest choice is a curated RefSeq transcript, because these sequences are manually reviewed and regularly updated. For TP53, we'll use this transcript. Before moving on, write down 3 pieces of information: the RefSeq accession number, the transcript variant, and the transcript length. Trust me, those 3 numbers will save you a lot of confusion later when you're validating your essay or writing your manuscript. Once you've selected a transcript, stick with it. Every step from this point forward should refer to this exact sequence. Now let's download the sequence we'll actually design primers on. Click on the transcript accession. You'll arrive at the nucleotide record. At first, the page looks overwhelming. You'll see annotations for exons, coding regions, UTRs, feature tables, and many other details. Fortunately, we only need one thing: the coding sequence, or CDS. A common question is, why not design primers using the entire mRNA? Because untranslated regions, the 5' and 3' UTRs, often differ between transcript variants. A primer designed inside a variable UTR might work perfectly for one transcript and completely fail to detect another. The coding sequence is usually much more conserved and therefore provides a much more reliable target. Now switch the display format to FASTA. This gives us a clean nucleotide sequence without annotations. If your FASTA contains a header line starting with the greater than symbol, don't copy that. Only copy the nucleotide sequence itself. Now paste that sequence into a text file and save it. We'll use this exact sequence throughout the rest of the tutorial. Before we start designing primers, there's one principle you absolutely need to understand. In my opinion, this single concept separates good qPCR assays from bad ones. Before we open Primer 3+, there's one rule you should remember throughout your entire career. If you forget everything else from this video, remember this one sentence: design your assay to amplify cDNA, not genomic DNA. Everything else is secondary. Let's see why. Genes are stored as genomic DNA. Genomic DNA contains both exons and introns. When a gene is transcribed, the cell first produces a precursor RNA that still contains both. During RNA splicing, the introns are removed and the exons are joined together. The mature mRNA and the cDNA synthesized from it contains only exons. Genomic DNA still contains all the introns. Now imagine your RNA preparation contains a small amount of genomic DNA contamination. This is actually very common, even after DNase treatment. If your primers amplify both cDNA and genomic DNA, the qPCR instrument has no way to distinguish between them. It simply detects amplification. As a result, your CT values become artificially lower, and your gene expression measurements become inaccurate. Fortunately, avoiding this problem is surprisingly simple. There are two standard strategies. Strategy 1. Design across an Exon-exon junction. Whenever possible, this should be your first choice. One primer, usually the forward primer, should overlap two neighboring exons. Part of the primer binds to one exon. The rest binds to the next exon. That exact sequence exists only in mature mRNA and cDNA. It does not exist in genomic DNA because an intron separates those two exons. As a result, genomic DNA cannot be amplified efficiently. Whenever an exon junction provides a suitable primer sequence, choose it. Strategy 2. Place primers in different exons. Sometimes the exon junction isn't suitable. Maybe the exon is too short. Maybe the GC content is poor. Maybe Primer 3+ simply can't generate a good primer there. That's perfectly normal. In those situations, place the forward and reverse primers in different exons. Let's look at an example. Here the forward primer is located in exon 3. The reverse primer is located in exon 4. In cDNA, those exons are directly adjacent. The PCR product is only about 100 base pairs long, perfect for qPCR. But in genomic DNA, those same exons are separated by a long intron. Instead of 100 base pairs, the polymerase would need to amplify several thousand base pairs. Under standard qPCR, conditions fragments that large amplify very inefficiently, or not at all. Again, the reaction strongly favors cDNA. Before leaving NCBI, scroll down to the Features section. Locate the XN coordinates. Write them down, or simply take a screenshot. We're going to use this XN map later. This is an important habit. Primer 3 Plus only evaluates thermodynamics properties. It doesn't fully understand XN boundaries. You do. And that's why biology should always guide software. Now that we know exactly where we want to design our assay, let's open Primer 3 Plus. Go to Primer 3 Plus. Paste the coding sequence we downloaded earlier into the sequence box. Before clicking Pick Primers, we're going to adjust several parameters. These values aren't arbitrary. They've been optimized over many years of PCR research and work well for most qPCR experiments. Let's go through them one by one. Find the primer size settings. Set minimum 18, optimum 20, maximum 25. Why 20 nucleotides? Because it's an excellent compromise between specificity and efficient binding. Very short primers may bind to unintended targets. Very long primers are more likely to form secondary structures and usually don't improve specificity very much. Around 20 bases is the sweet spot. Next, set the melting temperature, minimum 58 degrees Celsius, optimum 60 degrees Celsius, maximum 62 degrees Celsius. But here's something many beginners don't realize. The actual temperature isn't the most important value. The difference between the two primers is, after Primer 3 Plus generates the candidates, look at the TM of the forward primer, then compare it with the reverse primer. Ideally, the difference should be less than 1 degree Celsius. For example, 60.2 degrees Celsius and 60.6 degrees Celsius. Excellent. 60 degrees Celsius and 64 degrees Celsius? Not a good choice. If one primer melts much earlier than the other, they won't anneal efficiently during the same PCR cycle. Balanced primers usually produce much more consistent amplification. Now, set the GC content. Minimum 40%, optimum 50%, maximum 60%. GC pairs form 3 hydrogen bonds. AT pairs form only 2. Too little GC means weak primer binding. Too much GC makes the primer overly stable and increases the chance of secondary structures. Again, balance is the goal. Now, set the expected amplicon size. For qPCR, we generally want products between 80 and 150 base pairs. Short products amplify faster. They produce better reaction efficiency, and they generate much cleaner quantitative data. Long amplicons are perfectly acceptable in conventional PCR, but for qPCR, shorter is almost always better. Here's one setting I almost always change. By default, Primer 3 Plus returns only a handful of primer pairs. Personally, I increase that number to around 100. It only takes a few extra seconds, but it gives me far more options to evaluate. Remember, Primer 3 Plus doesn't choose the best primer. It simply generates possibilities. Choosing the best one is still your job. Now, click Pick Primers. Within a few seconds, Primer 3 Plus displays dozens of candidate primer pairs. Most beginners immediately choose candidate number 1. Don't do that. Now the real evaluation begins. Primer 3 Plus ranks primer pairs using thermodynamic scoring. It does not know your biological question. It doesn't know XN boundaries. It doesn't know whether a primer spans an XN junction. And it certainly doesn't know which transcript matters most for your experiment. So instead of blindly accepting the first result, let's evaluate the candidates systematically. For each candidate, look at 4 values: primer length, TM, GC content, amplicon size. If all 4 fall within the ranges we discussed, keep that candidate. If not, Skip it and move to the next one. Usually you'll narrow your list to just a few good candidates. Now we need to evaluate something Primer 3+ doesn't fully assess. A primer can be perfectly designed, have the right melting temperature, excellent GC content, and still fail in real samples. Why? Because one of the primer binding sites contains a common SNP. A SNP is a single nucleotide polymorphism, a naturally occurring variation in the DNA sequence. If a SNP lies inside your primer binding site, some samples will match the primer perfectly, while others will contain a mismatch. That mismatch can reduce primer binding, decrease amplification efficiency, or even prevent amplification entirely. The effect is especially severe when the SNP is close to the 3' end of the primer, because DNA polymerase begins extension from that end. So let's check our primers. Go back to the NCBI Gene page. Open the transcript we selected earlier. Open the NCBI Genome Data Viewer and enable the DBSNP. Locate the genomic region where your forward primer binds. Then examine the reverse primer binding site. Ask yourself one simple question. Does either primer overlap a common SNP? If the answer is no, great. Move on to the next step. If a common SNP falls within the last 5 nucleotides at the 3' end, I strongly recommend choosing another primer candidate, even if Primer 3+ ranked at first. Avoiding population variance usually leads to much more reliable qPCR assays. Remember, the goal isn't just to design primers that work in one sample. The goal is to design primers that work consistently across every sample in your study. Before ordering any primers, we need to answer one important question. Will these primers behave well during PCR? Primer 3+ evaluates many primer design parameters, but it doesn't show all the secondary structures we care about in an easy-to-interpret way. For that, we'll use another free tool, IDT OligoAnalyzer. Open the website. Copy the forward primer from Primer 3+. Paste it into the sequence box. Click Analyze. We'll evaluate 4 things: hairpins, self-dimers, heterodimers, GC clamp. Let's start with hairpins. A primer is supposed to bind to your template, not to itself. Sometimes a primer folds back on itself because parts of the sequence are complementary. This creates a structure called a hairpin. Every primer trapped in a hairpin is one less primer available to amplify your target. In OligoAnalyzer, click hairpin. You'll see one or more predicted structures. Don't panic if you see a hairpin. Almost every primer has some secondary structure. The important question is, how stable is it? Look at the delta G value. The more negative the delta G, the more stable the hairpin. As a practical guideline, delta G around -2 or -3 kilocalories per mole is usually not a concern. Around -5 kilocalories per mole, pay attention. More negative than about -7 kilocalories per mole, especially if the hairpin involves the 3' end, I usually reject that primer and choose another candidate. Now look at where the hairpin forms. If it's located near the middle of the primer, it's often acceptable. If the last few nucleotides at the 3' end are involved, that's much more problematic. Remember, DNA polymerase starts extending from the 3' end. If that region is trapped in a hairpin, PCR efficiency can decrease dramatically. So, when comparing 2 otherwise similar primers, choose the one with the weaker hairpin. Now click Self-Dimer. This predicts whether 2 copies of the same primer can bind to each other. At first glance, these diagrams may look confusing. Ignore most of the alignment. Instead, look directly at the 3' ends. That's the only region we really care about. If 2 primers bind strongly through their 3' ends, DNA polymerase may extend them. Instead of amplifying your gene, it starts amplifying primer dimers. Those primer dimers compete with your target for regions. They consume primers, They consume nucleotides, and they often appear as extra peaks in melt curve analysis. So what should you look for? If the complementarity is mainly in the middle of the primer, it's usually acceptable. If the last 3 or 4 bases at the 3' end pair perfectly, that's a warning sign. Again, when comparing candidates, choose the primer with weaker 3' interactions. Now let's evaluate the interaction between the forward and reverse primers. Copy both primer sequences into OligoAnalyzer. Click heterodimer. The interpretation is almost identical. We don't care if the primers weakly interact somewhere in the middle. What worries us is strong complementarity at the 3' ends. Why? Because now the forward primer becomes the template for the reverse primer. The reverse primer becomes the template for the forward primer. And the polymerase happily amplifies those dimers instead of your target. If 2 candidate primer pairs have similar thermodynamic properties, always choose the pair with weaker heterodimer formation. There's one final feature to check before leaving OligoAnalyzer. Look at the last 2 or 3 nucleotides at the 3' end of each primer. Ideally, 1 or 2 of those bases should be G or C. This is called a GC clamp. Why does it matter? GC base pairs form 3 hydrogen bonds. They're slightly more stable than AT pairs. Having 1 or 2 G or C bases at the very end helps anchor the primer exactly where DNA polymerase begins extension. But don't overdo it. 3 or 4 consecutive Gs or Cs at the 3' end can actually increase non-specific amplification and primer-dimer formation. So the goal isn't to maximize GC. The goal is balance. 1 or 2 GC bases at the 3' end is usually ideal. At this point, many people stop. Don't. Now it's time to compare your primers with the XN map you saved earlier. Go back to the transcript record. Locate the XN boundaries. Now ask yourself, where do my primers bind? If one primer spans an XN-XN junction, excellent. If not, are the forward and reverse primers located in different XNs? Also excellent. If both primers are located entirely inside a single XN, be careful. That design is much more likely to amplify contaminating genomic DNA. Even if primer 3+ ranked it first, biology is more important than software rankings. This is why experienced researchers don't simply accept candidate number 1. They compare the biological context as carefully as the thermodynamic parameters. There's one final step before ordering your primers, and in my opinion, it's one of the most important. Even a perfectly designed primer is useless if it amplifies the wrong gene. To verify specificity, go to NCBI Primer Blast. Paste your forward and reverse primer sequences. Choose your organism. For today's example, we'll select Homo sapiens. Then click Get Primers or Search. Primer Blast aligns your primers against the entire genome. This tells you whether your primer pair amplifies only your target, or whether it also matches other genes. The ideal result is simple. 1 predicted PCR product. 1 target gene. No significant off-target amplification. If Primer Blast predicts multiple products, go back to Primer 3+ and choose another candidate. Never skip this step. Ordering a new primer pair costs money. Running another PCR experiment costs time. Spending 2 extra minutes on Primer Blast is almost always worth it. Before clicking order, run through this checklist one last time. Transcript correctly selected. Correct coding sequence downloaded. Primer length between 18 and 25 nucleotides. TM, around 60 degrees Celsius. Forward and reverse TM differ by less than 1 degree Celsius. GC content between 40 and 60%. Amplicon size between 80 and 150 base pairs. No common SNPs in primer binding sites. Weak hairpins. Weak self-dimers. Weak hetero-dimers. Appropriate GC clamp. Primers span an exon junction, or are located in different exons. Primer blast confirms specificity. If every box is checked, you're ready to order your primers. Today we've gone through the complete workflow for designing qPCR primers from scratch. In the next video, we'll move one step further. We'll design a TaqMan probe, discuss fluorophores and quenchers, and learn how to build a highly specific hydrolysis probe assay from the primers we've designed today. If you found this tutorial helpful, consider subscribing, because the next video completes the entire qPCR assay design workflow. If you're working on a research project and need support with qPCR assay design or other bioinformatics analyses, visit bioinformatics.ca to learn more about our services and how we can help with your research. Thanks for watching, and I'll see you in the next video.